I am Aruna Punna, working as Junior Lecturer in Botany, Government Junior College for Girls, Bongir. Today our topic is Microbes in Human Welfare. In this class, we are going to learn about microbes in industrial products. Under this topic, production of antibiotics, chemicals, enzymes, and other bioactive molecules. Microorganisms are used in the production of antibiotics. In 20th century, different types of antibiotics produced for the human health welfare. And in Greek language, what is antibiotic? Anti means against. Here against means against the disease causing organisms. But related to, with reference to human beings are to live, not against. Antibiotics used only against for the disease causing organisms, not for the human beings. Human beings want to live and kill only disease causing microorganisms. And antibiotics are one kind of chemical substances which are produced by different types of microorganisms and kill disease causing microorganisms. The first familiar commonmost antibiotic is penicillin. This is unexpectedly discovered by Alexander Fleming. He is a microbiologist. When he was working on Staphylococcus bacteria, he discovered this penicillin antibiotic. Generally, when we suffer from any infection, so what we do? We go for a diagnostic center, laboratory, and we give the infected sample to the laboratory person. The laboratory person take that infected sample and by culturing method, this is petri dish, by pouring nutritive media, on the nutritive media only he grow the infected sample. Then after 1 week, infected sample microorganisms are present. So these microorganisms grown into colonies. Then by observing these colonies under microscope, the lab technician identify disease. That is why by identifying that particular microorganism, then he give the report to us. So in the same way here, Alexander Fleming also microbiologist. Then he was working on Staphylococcus bacteria. This bacteria generally cause infection on wounds. So one day he observed a mold growing on unwashed culture plates. Actually, after completing their research, they clean immediately the culture plates. But one unwashed culture is like that only. So on that unwashed culture, one mold growing he observed. What he observed, around the mold, Staphylococcus bacteria is not growing. See here, this is petri dish. This is original picture what Alexander Fleming observed. These are the colonies of the Staphylococcus bacteria. This is one mold. Around this mold, Staphylococci undergoing lysis, means gradually they are dying. Then he observed this mold. Mold means fungi. This fungi is producing one kind of chemical. By the activity of this chemical only, these Staphylococci bacteria are dying. Then finally, he gave that chemical as penicillin because that mold name, that fungi name is Penicillium notatum. So in this way, here Penicillium notatum is producing the chemical, that is why the name has been given to the chemical. And after many intensive researches, finally a effective penicillin antibiotic was established by Ernest Chain and Howard Florey. This antibiotic extensively used to treat American soldiers, those who are wounded in Second World War. For this discovery, great, excellent discovery, Alexander Fleming, Ernest Chain and Howard Florey awarded the Nobel Prize in 1945 after Second World War. And after penicillin also, different types of antibiotics discovered, purified from different microorganisms. The world suffered from and suffering from different types of diseases. In previous days in the world, the plague, whooping cough, diphtheria, leprosy, very dreadful. Now like we are all are suffering from corona, like that. At that time also, these diseases are very dreadful, sorrowful, and lakhs of people died due to these diseases. So, which are caused by harmful bacteria and kill millions of people all over the world. Small information I will give you about these diseases. Information about plague. The common symptoms of plague, fever, weakness and headache. This disease spread all over the world. In common era 541 and 1347 and 1894, many people suffered from this disease. And now this disease can be prevented by vaccine, and we can treat this disease by using antibiotics like gentamicin and fluoroquinolone. And information about whooping cough. The other name is pertussis. Generally, this disease we can see in infants. In infants, respiratory tract infected. In 2012 in America, nearly 48,000 cases occurred. And now this disease can be prevented by vaccine and treatment by using antibiotics like azithromycin and clarithromycin. And information about diphtheria, the common symptoms are nose, throat infection, and lower jaw, lymph glands swelling, fever, weakness. In 1735 in England and 1826 in America and Europe, most of the people suffered from this diphtheria. At the time, the mortality percentage is approximately 40%. Means, for example, 100 people are affected by this diphtheria. Out of 140 people died. So, very dreadful and sorrowful condition at that time. And now this diphtheria disease can be prevented by some vaccine, and we can treat this disease by using antibiotics like erythromycin and penicillin. After so many researchers, this is triple vaccine. By giving one vaccine, 3 diseases can be prevented. That is DPT vaccine. D means diphtheria, P means pertussis, other name is whooping cough, and T means tetanus. These are all bacterial diseases. Tetanus bacteria generally enters through the wounds and cause muscle contraction disease. That is why when we get any injury by the iron, immediately we go and take tetanus TT injection. So TT injection is for to stop the tetanus bacteria. Then information about leprosy. The general common symptoms are lesions on the skin and damage of nervous system, and sometimes leads to blindness also. This leprosy somewhat can be prevented by using BCG vaccine. Actually, this BCG vaccine prepared for the treatment of tuberculosis, and we can treat this leprosy disease by using antibiotics like dapsone, rifampicin, clofazimine. And in India, to eradicate completely this leprosy disease, National Leprosy Eradication Program is going on. This is established. Here you can see throughout the world per year, 2 lakhs of people are suffering from leprosy. Out of 2 lakhs cases, 1.25 lakhs people are from India. That means approximately 59% cases are from India only. Even though National Leprosy Eradication Program is going on, such large cases are coming from India. This is somewhat sad. We could not imagine the world without antibiotics. And the chemicals, enzymes, and other bioactive molecules also produced by the activity of the microorganisms. Here, what are the chemicals? So here, first, Aspergillus niger, this is one kind of fungi. It produces citric acid. Acetobacter acetate, a bacterium which produces acetic acid. Clostridium botulicum, it is a bacterium which produces butyric acid. Lactobacillus, it is a bacterium which produces lactic acid. All these citric acid, acetic acid, butyric acid, lactic acid used in food and beverage cosmetic industries. In food generally for the storage purpose and to give some flavor to the food. And in the beverages means in the fermentation process. Beverages also to add some flavor, to increase some taste. And cosmetic industries, water cosmetics means any lotions which are applied to the face, body lotions, shampoos, lipstick. So in all productions, these acids are used. Yeast, Saccharomyces cerevisiae, you know all. This is used in the fermentation process used for ethanol in beverages industries. Already we learned about this. And so these are all comes under chemicals. Now enzymes. Enzymes are 3 enzymes are there: lipases, pectinases and proteases, and streptokinase. One by one I will explain. Here lipases. These lipases generally used in the preparation of detergents which are helpful in removing oily stains from laundry. These lipases clean our cloths in this way by removing oily stains. And pectinases and proteases. These enzymes, bottled fruit juices, are clarified. Here I will explain this one. This is apple juice. This is also apple juice. But this is homemade. This is industrial made. See the difference. Here industrial made juice is very clear and transparent. But here homemade juice is very thick and pulpy. What happens in the industries at the time of juice preparation? These proteases and pectinases added to the juice. So these enzymes completely digest the pulp. That is why finally this clean and transparent juice can be get. And next one is streptokinase. This is one kind of enzyme produced by Streptococcus bacterium. What is the use of this streptokinase? So here, this is heart. These are the blood vessels which are supplying blood to the heart. So here, this is the magnifying view, blood vessel artery. These are the walls. This is the inside lumen. Through this lumen, blood is flowing. But here what happened? Some clot due to deposition of bad cholesterol, clot is formed. So this clot is obstructing the blood flow. So what happens? Immediately blood flow is stopped to the heart muscle. Immediately, due to stopping of blood supply to the heart muscle, this heart muscle damage. At that time, person get heart attack. So immediately we have to take the heart patient to the hospital. At that time, doctors immediately give one injection. That injection has streptokinase enzyme. This streptokinase enters into bloodstream and immediately dissolve this clot somewhat, mostly. So in this way, we can stop the death of that person. So this streptokinase used as clot buster for removing clots from the blood vessel of patients. So here myocardial infection means heart muscle is damaged. That is why this condition is leads to heart attack. Here this streptokinase enzyme produced by Streptococcus bacteria and modified by genetic engineering. What is genetic engineering? Procedure of genetic engineering technique. This technique you can learn in biotechnology lesson in detail, but here simply I will try to explain you. First we have to take one bacterium. This is genetic material of bacterium. This is plasmid. We have to separate this plasmid, and this plasmid is made up of DNA and ring-like structure. It can replicate itself. So due to this replication capacity only, here we are using this plasmid. Here plasmid is ring-like structure. We have to cut this plasmid by using restriction enzymes. These enzymes cut the plasmid wherever we require. Then, so ring-like structure opens. Then here you have to select particularly streptokinase gene. Here our ambition is to produce streptokinase enzyme in industry. Each gene is responsible for one activity. Here this gene is responsible for the production of streptokinase. This streptokinase gene we have to isolate from the Streptococcus bacteria, and this streptokinase gene inserted into this plasmid, then fusion formation takes place. This fusion by the activity of ligase enzyme completed. So here this plasmid is having streptokinase gene. Now this plasmid can produce streptokinase enzyme only. Directly we can use Streptococcus bacteria for the production of streptokinase enzyme, but when we want to use this genetically modification method, what happens? Directly when we take Streptococcus bacteria, that bacteria can produce so many enzymes along with streptokinase. But here we need only streptokinase enzyme. That is why we have to particularly separate this streptokinase gene only, and then we have to insert into this plasmid. Then after completing the genetical modification, we have to insert this plasmid into the bacterium. Now this bacterium called as genetically modified bacterium. Now this bacteria can produce streptokinase enzyme only. This bacteria never produce any other enzyme because it is having only one streptokinase gene. Then this bacteria goes on multiply. After so many multiplication, the number increases. So all these bacteria start producing streptokinase protein, and these bacteria grown in a fermenter. After completing the fermentation process, this streptokinase protein undergo purification process. After purification, this enzyme sealed in small bottles. These small bottles transport to the hospitals. This medicine only given to the heart patients at the time of heart attack to protect the patient. Here, two types of bioactive molecules I will discuss here. One is cyclosporin A and statins. This cyclosporin A is a immunosuppressive agent. At what time this medicine has to be given to the patient? Here I will explain this by taking one example. Here one person is suffering from kidney problem. His two kidneys are completely damaged, not working. That is why he need functional good kidney. Immediately he need kidney transplantation. So that is why we have to collect kidney from donor. The donor may be live or may be brain dead. So from this donor we have to collect kidney, and this kidney transplanted into the patient by surgery. After surgery, generally doctors prescribe the patient to use this cyclosporin A medicine. It is immunosuppressive agent. What is its activity? Generally, what happens? Any disease-causing microorganisms are foreign bodies. So, when any foreign body enters into our body, what happens? Immediately, our immune system recognizes that foreign body and kills that. So, here also, in the case of this patient, here we transplanted kidney. So, his immune system recognizes the transplanted kidney as foreign body. So, his body won't accept this transplanted kidney. The body rejects this transplanted kidney. That is why temporarily we have to stop his immune system by giving this cyclosporin A medicine, immunosuppressive agent. So this is used in organ transplant patients. This cyclosporin A produced by Trichoderma polysporum, it is a fungus. At the same time, when the patient has been given this medicine cyclosporin A, within 6 to 12 months, his body accepts this newly transplanted kidney, and at the same time he has to follow proper hygiene. Otherwise, already in his body, by using cyclosporin A, his body don't have immune system. At the same time, if the person won't follow proper hygiene, his body infected by so many infections, and that infection may lead to the infection of transplanted kidney also. This may lead to even death. So in many cases, even though the organ transplantation surgery is successful, but not maintaining proper hygiene due to infections, the patient may die. So in this way, using medicine and maintaining a proper hygiene is very important. Then only we can save the patient. So cyclosporin A produced by Trichoderma polysporum A fungus. Then statins. These are one chemical substances which are produced by yeast, that is Monoscus purpurus. What is the activity of this one? Here, when we eat overfood, the extra food converted into cholesterol, that cholesterol may be deposited under the skin. That condition leads to obesity, or the cholesterol enters into the bloodstream, leads to heart attacks. So, we have to take limited food. That extra food, what happens? In the biochemical reactions, the extra food converted into HMG coenzyme A, beta hydroxy beta methyl glutaryl coenzyme A. Generally, this HMG coenzyme A converted into cholesterol in the presence of HMG coenzyme A reductase. So, this is enzyme. This is active site of the enzyme. So, HMG coenzyme A is a substrate. This substrate enters into the active site, biochemical reaction completed, and cholesterol is formed. Here, by using these statins, we can stop the cholesterol formation. In which way? This I explained in the enzymes lesson, competitive inhibitors. In that topic I explained. So, here what happens? These are the statins. The statins molecular structure is same as HMG coenzyme A. So, here these statins act as competitive inhibitor and reach the active site of HMG coenzyme A reductase. So, when these statins occupy this active site, so what happens? There is no active site for the HMG coenzyme A. So, HMG coenzyme A could not undergo biochemical reaction and could not convert it into cholesterol. In this way, these statins lowers the cholesterol level, can act as a competitive inhibitor, and in this way, the cholesterol synthesis stopped. These medicines generally prescribed by the doctors to heart patients. In the next class, I will continue this lesson with microbes in sewage treatment, microbes in production of biogas, the structure of biogas plant, microbes as biocontrol agents. The 2 marks and 4 marks question and answers of this lesson, English medium, are in the description. If you like this video, please share and subscribe to my channel. Thank you.